Hello everyone, it's me Rachit back here in another new video. Before we get started into the next disrupting technology, I wish you guys a very happy Diwali and hope you guys had a very great time. So what is CRISPR? So the next disrupting technology is CRISPR. So CRISPR stands for clustered regularly interspaced short palindromic repeats. It's a very big word, but it's very easy to understand. So it's an innovative genome editing. It edits and changes a gene so it can cure diseases or we can act differently. And it also addresses the cure for genetical disorders like sickle cell anemia, retina pigmentosa, and also cancer. I mean, cancer is not official yet, but scientists are experimenting with CRISPR to cure cancer. So and also improves agriculture practices, so it improves the quality of the plants in agriculture so it can be more stronger and it can fight the pests, diseases, and bacteria. So we need to know what are the tools of CRISPR, right? So the tools of CRISPR, there are three tools: the cas9 protein, the guide RNA, and the delivery systems. So the cas9 protein, it's an enzyme. The cas9 is an enzyme which acts as a molecular scissors, which basically acts like a scissor. We all know what a scissor does, right? It cuts something. So the cas9 is a scissor which cuts the DNA so that scientists can edit that part of the DNA and change the part of the DNA. So the second thing is guide RNA. So, Cas9, okay, it edits the DNA, cuts the DNA, we know that. But how does it know where to actually go in the DNA so it can edit? So that's where guide RNA comes into help. So this RNA molecule, so the guide RNA molecule matches with the DNA sequence in our body, so it can direct and guide the Cas9 into our DNA so it can edit. So it basically matches with our DNA sequence. So it basically knows everything about our DNA and directs the Cas9 into our DNA so it can edit. So together, these two together is called the CRISPR tool. So how does this CRISPR tool actually get into the cell? Okay, guide RNA knows about the DNA, but not in the body, the cell, and everything. So how do they know where to get how to get into the cell? So the delivery system, such as plasmids and viral vectors, take the CRISPR tool into the cell so they can do their work. So yeah, these three interconnect and work together so that they can edit and make changes in our body and DNA. So now we know the tools and everything, right? But how does it actually work? How does it work in the micro level? So before we get started, before we edit the DNA or gene, we first need to know the target gene. We first need to know which part of the gene we need to edit, so that like we can't just edit for fun, right? We have to know which gene we have to edit. So it can be a faulty or unwanted gene causing a problem. So after we know the target gene, we have targeted our gene, we know which gene to edit. We have to design the CRISPR tool. So the guide RNA, right? It matches with our DNA sequence. After we design the guide RNA, we attach the Cas9 enzyme. I already told what it's a Cas9. It's a scissor which cuts the DNA and edits it. So then we attach the Cas9 to the RNA, to the guide RNA. So these both form the CRISPR tool. So first we have found our target gene, we have created the CRISPR tool. So now we have to make everything work. So then we find the DNA and then we cut the DNA. So after cutting the DNA, the cells in the DNA automatically repair the DNA because it's been cut. So our natural automatic repair system tries to repair the DNA. So that's when the editing part comes in action. So when the natural repair system, the cells are trying to repair the DNA where it's been cut, then scientists can use this as an advantage and then add or remove or fix genes in that particular time and that particular area. So that's how the DNA is edited. It's very interesting, right? So we know how CRISPR works, but where does CRISPR come in action? Why should we know about CRISPR and everything? So the uses of CRISPR. So CRISPR, comes in handy in medical applications, agriculture advancements, and biological research. So, medical applications. It treats genetical disorders like sickle cell anemia, cystic fibrosis by correcting defective genes. So, the sickle cell anemia, I'm just going to explain this briefly. So, it is sickle cell anemia. Sickle cell anemia basically it's a red blood disease. Okay, it's a red blood disease, red blood cell disease. So, our normal red blood cells are round, flat, and flexible, or it's like a disc. But the sickle cell people with sickle cell anemia, their red blood cells are shaped like a sickle, like a cryosant, like a banana, basically. So this wide, long shape basically blocks the blood vessels, which causes organ problems, organ failures, and you know, like improper deliver of oxygen to the parts of the body. So why is sickle cell anemia actually caused? It's caused in the HBB gene. So the HBB gene is with hemoglobin. Hemoglobin is a red blood cell which delivers oxygen to the parts of the body. So the HBB cell, it's caused by a mutation. Mutation means change in the gene. So because of this change, that HBB gene is producing abnormal hemoglobin, not producing proper hemoglobin, which is causing this entire problem. So how does CRISPR come in action in the sickle cell anemia? So CRISPR basically edits and changes, it repairs the HBB gene so it can produce proper hemoglobin so that hemoglobin can work properly and the sickle cell anemia problem won't come again. So yeah, the next use of CRISPR is agriculture advancement. So as I said, CRISPR, what do you call it, edits the plants genes so it can be more stronger and can fight pests. So yeah, it won't ruin the plants, and it properly produces food. So the next use of CRISPR is biological research. So yeah, using CRISPR we can study about how gene functions and make new discoveries about the gene. So yeah, those are the uses of CRISPR currently, but there are going to be many more uses. So as I said, any good thing comes with a bad thing as well. So there are three main challenges of CRISPR. So I'll go through all of them. So the first one is off-target effect. So CRISPR may sometimes edit unintended parts of the genes. So it sometimes may edit parts which we don't want to edit. Sometimes it like edits a good part of the gene which we don't want to edit, leading to many complications and consequences, which is a mess. And also ethical concerns. So the ability to manipulate genes, the change the genes raises ethical questions like, we are manipulating human lives, you're changing human lives, and we're changing how they live. So it also raises ethical questions in the society as well. So regulatory hurdles is a very important one because like government keeps some rules, laws, and certain stuff which scientists and companies must follow before using CRISPR or releasing CRISPR based products, especially in humans, animals, and etc. So thank you everybody for joining me in this session. Hope you guys understood about CRISPR, and stay tuned for the next disrupting technology. Until then, it's me, Rachit, signing off. Bye bye.